Thank you for the invitation, and I'm honored to present the Onco Network consortium, which is formed by 8 laboratories, one in UK, one in Dublin, Ireland, 2 in France, 2 in Italy, and 1 in Switzerland and 1 in the Netherlands. The goals of this Onco Network has been to develop an Ion AmpliSeq cancer panel related to colorectal and lung cancer. We selected 22 genes that are involved in this disease and have meaning not only in the pathogenesis but also to a potential response to therapy, and defined the target sequence for these 22 genes. We ended up with 87 amplicons with a length which is in average 160 base pairs because we wanted and we want to work on paraffin tissue. Now, the work plan was on 3 steps. Phase 1, first 5, each lab performed the same experiment on the very same 5 paraffin-embedded samples. The second phase requires 10 colorectal and lung cancer that are sent blind in a paired fashion. And then the third phase, each lab performs 15 different formalin-fixed paraffin-embedded samples. Now, the 5 samples that were the controls were all had KRAS mutation, which is a basic, and then 2 of them also had the PI3K mutation. Now, each of the 4 labs who participated at the very beginning found any mutation. Also, an additional one in FBXY7, which was not described at the beginning in these samples. But no one could find a PI3K mutation that was described inside 2 of these samples. The problem was that there is a highly conserved homologue elsewhere in the genome, and then lowering the filtering of the software reading, actually we could find it. I mean, you can see on the upper side the germline variants or a mutated one, but on the right side where you see the second arrow, there is the confusion made up from the pseudogene. But actually, when we lowered the reading, modifying a little bit the software, everybody could find the mutation. It was only a small problem of software. So this means that the first step was passed and we passed the second one, and up to now 3 laboratories have finished the work, and each lab has found the very same sequence that were found by the first lab. So 8 on 8 in the case of lab A, 7 on 7 variants found in these samples by lab C on samples sent by lab D, and so on. This means that actually we are on the way to have developed a panel workflow and also a data analysis using the Ion Reporter software, which is very sound. And up to now, we found 100% concordance. So we are on the right way to have a good panel that may help us to make applied research in view of some real application for the patients. Thank you very much. This is on behalf of Professor Puig, of course. He prepared the slides, so I used my voice for his presentation, and we have to thank him, actually.